I would like to introduce our first speaker, Lincoln Nadauld, who's from Intermountain Health. Come on up, Lincoln, and we'll get you started.
Thanks, Mia, and I just want to thank the organizers for the opportunity to be here at this workshop. I thought yesterday's sessions were outstanding, very informative. The speakers were terrific. All of the panel conversations were really useful and informative. And today, I'm excited to talk a little bit about our experience at Intermountain Healthcare in implementing precision medicine and specifically precision oncology, and how we have gone about trying to measure the outcomes for patients and really try to measure value as we implement precision medicine, which is a whole separate part of how you go about using precision medicine is measuring clinical utility. Here are some of my disclosures, all of my disclosures. For those who may not be aware, Intermountain Healthcare is an integrated health delivery system headquartered in Salt Lake City, Utah. It's comprised actually now of 23 hospitals. There's a medical group, so we have employed physicians, and there are also affiliated physicians. And then there is a health plan called SelectHealth that now covers about 1 million patient lives. That's not the entire state. There are nearly 4 million people in the state. So SelectHealth covers a fairly large percentage of patients in the state. On an annual basis, Intermountain Healthcare sees 50% of all of the patients seen in the state of Utah. So we touch a lot of people there and have records on a lot of those patients. While SelectHealth covers a significant percentage, the rest of patients are covered by other health plans, United, the Blues, et cetera. Another important aspect of Intermountain Healthcare is that unlike some other integrated health systems, it is not a closed system, meaning patients don't have to have the SelectHealth health plan to be seen there, and physicians don't have to be employed by the system to practice in Intermountain Healthcare facilities and hospitals, so it's an open system. When we first implemented precision oncology, we started actually about 5 years ago. We built a workflow that looks like this, and I've seen similar workflows at conferences all over the place, and I've been lobbying in other settings to have us all just use one workflow slide because I think it's a pretty similar workflow. And this is the one we've come up with. I'm happy to share it with anybody. The major points that I'll comment on are this molecular analysis portion, the next generation sequencing portion. When we first built our precision oncology program, we had a big debate about whether to outsource this or whether to build it ourselves in-house. And ultimately, we won and built it in-house. And that debate included things like, we want to control turnaround time, we want to control costs, and we want to control all of the primary data. At least those are the arguments that we used for why we should build it in-house rather than outsourcing to one of the many commercial labs that offer this. I'm not sure that with all of these scars and trauma that I've been through 5 years later, I'm not sure that's the right decision for every health system. But it was the right decision for us, and it has been really beneficial. And so there is a lot of upside to having this in-house and being able to do it in-house. And increasingly, it is doable. There are enough partners in the space to help you pull it off. We have a targeted sequencing panel that is 165 genes. That sees all of the relevant mutation types, and we're doing somatic testing only. At least when we started, we were only doing somatic testing. We weren't doing tumor normal pairs. So it gives you a little flavor for how we set it up. Then the other point that I'll comment on in this slide is this molecular tumor board portion. When we first launched our program, we quickly found that physicians would order the test, but when we tried to measure who was acting on it or how it changed management, we found that it very infrequently changed management. Less than 20% of the time did physicians use that information. When we probed on that deeper, we found that the primary reason is that they felt uncomfortable interpreting the data. They weren't sure what this long list of genes meant, outside of BRAF and EGFR and maybe KRAS, most of our frontline medical oncologists did not feel comfortable interpreting additional genes. And so these next generation sequencing reports can read like Greek to most of our frontline colleagues. So to overcome and try to enable precision oncology a little further, we established a molecular tumor board that is internal but includes external experts. And it meets and actually reviews every single test ordered and performed in our system. And then we put the molecular tumor board interpretation right on the report so that the frontline provider can see that, and we hope that it distills down for them the salient points. And so what's represented here in this slide is an example of one of our reports where what is listed for the doc is just the gene and some drugs they can consider, and these drugs are actually listed in priority according to how the molecular tumor board would use them. They would say, so we start with this, first drug and move to the second drug. The additional information is available on the report, so if a provider wanted, they could scroll down and see the exact gene and the exact alteration and the nucleotide substitution and all of those things, and they could see all of the VUSs, et cetera, but that's on page 2. So that what they really want is right there on the front page. And then we actually even included right in this system an order button so that if the doc liked the gene drug match that they saw, they could even go ahead and order that drug, and we would engage a drug procurement team to help obtain that drug. To measure whether this precision oncology approach was effective for patients, we performed a study internally where we looked at patients who had received genomic testing and received targeted therapy, and we compared those patients on a case-by-case basis to matched control patients also treated within our system who had not received genomic testing and had not received targeted therapy but had gone on to receive the next line of therapy. We matched these patients according to age, gender, diagnosis, and number of previous lines of treatment. So a matched pair of patients, a case and control set would be 2 patients, 65-year-old men with metastatic pancreas cancer or metastatic lung cancer who had both been through 2 previous lines of treatment, for example. And then we looked at progression-free survival and ultimately overall survival. And we looked at the cost of care, which was a very big question in our health system. The payer side wanted to know what this was costing, and the provider side wanted to know as well. And honestly, the executive leadership team was really curious about what this was costing as they initially were funding this effort. So this is one of my favorite slides to show because we attempted in one figure to show not only survival, and just to comment on that, we found that those treated with standard therapy had about a 26-week overall survival. And keep in mind, these were patients who had been heavily pretreated. On average, they had received 3 previous lines of treatment. When we looked at the precision oncology group, they had a much longer overall survival and nearly a doubling of the overall survival. What's shown in these hatched areas are charge events. So every time there is a yellow or red little box, that means there was a medium or high charge event in that patient's care. And green boxes are lower charge events. And so you can start to kind of compare what the overall sort of heat map looks like as charge events for patients. And each gray bar is a patient. And you can see, you can start to draw conclusions about the intensity and frequency of charge events in that patient's overall care. Now, no one can add all that up and come to a conclusion, so we did it for us. We looked at all of the numbers, and when we took into account the entire cost of care, including outpatient costs, inpatient costs, the cost of drug, the cost of testing, and everything, we found that overall the cost was actually a little lower in the precision oncology group. In fact, we saved $730 per patient. Now, we normalize this to week of survival. So this is $730 saved per patient per week of survival. And we did that because, of course, the patients in the precision oncology group lived twice as long. So their overall cost over that extended lifetime was larger, but per week of survival it was lower. When we tried to look at a larger population of patients, we took almost 2,000 patients from our SelectHealth population who had received, we looked at 100 who had received targeted therapy and another 1,000 who had not. And we looked just at the last 3 months of survival, which is a big deal for many payers. They want to know what happens at the end of life. And we found that patients actually were having fewer inpatient costs, and actually the total overall cost in the last 3 months of life was lower in the patients who received this precision oncology approach. And this was across several thousand patients. Interestingly, the outpatient costs, and as would be predicted, the drug costs or pharmacy costs were much higher in the precision oncology cohort, almost 300% higher for these targeted therapies than whatever therapy patients on standard care were receiving. However, we made up for a lot of those costs by saving on inpatient hospitalization events. The side effects, et cetera, seem to not require as many inpatient hospitalizations for those patients. And I think there were other factors involved that we're still exploring, things like patients on precision medicine just being able to spend more time at home. And I think there may have been some selection bias in patients who are well enough to be receiving treatment compared to those who were in the control group. And so we're still sifting through some of that. On the strength of this improved survival and decreased costs, we talked to our health plan about changing their policy, and ultimately they were sufficiently impressed by the data that SelectHealth has now changed its policy to cover payment for this kind of molecular testing for next-generation sequencing in patients with advanced cancer. So that was very encouraging for us, and we decided to pursue this model and expand now our precision medicine efforts. And we have gone into other aspects of precision medicine, in some cases even outside of oncology. And we have these tests that we're now offering in our next-generation sequencing lab. The first that I mentioned is this targeted somatic testing panel called ICG-100, where we look at 165 genes. And this is the test that our health plan, SelectHealth, is covering for advanced cancer patients. We're going to use the same model where we go and test a cohort of patients, closely monitor their outcomes, look at the financial and economic impact, as well as the patient outcomes impact, and then share that data with SelectHealth to discuss whether these additional tests should be covered. So a couple of the additional tests are this pharmacogenomic test for mental health and our behavioral health and primary care clinics for patients who are receiving a new diagnosis of depression or anxiety and will be starting new antidepressants or anti-anxiolytics. We're actually rolling that out through our clinical program. And then we have an inherited germline testing panel that we'll be using starting in January in all newly diagnosed cancers of all stages. And we're going to take the same approach where we initially will not bill for that, and in the first 500 patients, we'll just use that as something of a pilot project and monitor the outcomes. And if we see a positive impact, then we'll talk to our health plan about covering. A couple of things that I'll just mention as I wrap up here. One way that we're really informing our efforts in precision medicine is by using this biorepository that consists of about 5 million samples that have been stored since 1975. And in addition to all of these samples we have in our biorepository are all of the electronic outcomes associated with each one of those cases. And that can be very powerful, especially if we're able to liberate all of the genomic data. And so we have built a high-throughput sequencing center. And this gives you a flavor for the kinds of patients we have in our biorepository. It's about 200,000 now oncology specimens. These are mostly FFPE wax blocks with the annotated clinical outcomes. And they are distributed according to how you would predict based on the relative incidence of various cancers. We have other kinds of specimens in there, including CNS and cardiovascular related samples, et cetera. And so we have launched now this high throughput sequencing center where we have capacity to do about 20,000 whole genomes per year, and we're now in the process of pulling samples out of our biorepository, doing the whole genome analysis, combining that with the clinical outcomes, and then making predictions about how to treat future patients based on what has happened to past patients, and using a variety of AI tools to better understand and characterize these different patient cohorts. So with that, I'll just say thank you for the opportunity to present and thank all of the people on this slide. And I think, are we doing questions as part of the panel? Yeah, okay. Thank you.